This lesson, Molecules to Metabolism, is an introduction to biochemistry. Here are your objectives. So to begin talking about biochemistry, we need to start by talking about carbon. Carbon has 4 valence electrons, or outer shell electrons, and that allows it to form 4 covalent bonds. A covalent bond being a chemical bond where one atom shares its electrons more or less equally with another atom. And carbon is great at doing that. So it can form 4 covalent bonds, which I'm showing here by these 4 sticks. That allows carbon to form long chains that can form a skeleton or a backbone onto which we can add other things. And that's why it's central to the chemistry of life. Carbon can form these long chains. Those chains can be branched as well, and that allows an even greater variety in the kinds of compounds we could create. Also, there could be single or double bonds in those chains, and that further increases the variety. And in fact, the chains can even double back on themselves, forming rings. So it's because of this ability to form so many different kinds of structures that carbon is central to the chemistry of life. When we talk about organic chemistry or organic compounds, we're using the word organic differently than you do in the grocery store, where it usually means no pesticides. An organic compound is simply a carbon-containing compound, with the exception that we don't include carbon dioxide in that. We don't call carbon dioxide an organic compound. And so organic chemistry is the study of carbon-containing compounds. When the chemistry of living things was first being studied, it was felt that there must be something necessarily special about them because they came from living organisms. And central to this idea was the hypothesis of vitalism, or the vitalist hypothesis, which held that living things contain a special life force, and that made them different from non-living things, and that in fact you couldn't study biological processes by breaking them down to purely physical or chemical processes, purely kind of mechanical processes, because there was this extra special force that biological things had that non-living things didn't. And following from that was the idea that only living things could manufacture biological compounds, because these were special compounds imbued with this life force that differentiated the living from the non-living. Now, part of the history of the overthrow of the vitalist hypothesis was the synthesis of urea. So urea is a molecule that looks like this, that contains both carbon and nitrogen, as well as hydrogen and oxygen, and urea is actually a compound that's present in urine, hence the similarity in the name, made in the liver from amino acids. And it's from these amino acids that this nitrogen comes, and it gets attached to carbon to get rid of it. So this is part of how our bodies get rid of excess nitrogen from amino acids. Now, in 1828, German chemist Friedrich Wöhler actually artificially synthesized urea. And because he had artificially synthesized this, not using any aspect of a living organism, he had shown, therefore, that you could make a biological compound, something that was previously thought only to come from living things, in the lab. And that helped to erode the idea of vitalism. And as more and more compounds like this were made in the lab, eventually vitalism was left behind. Let's shift gears a little now to talk in general terms about how these carbon-containing compounds are built up and broken down. So as we've seen before, metabolism is the sum of all the chemical reactions in a cell or in an organism. Or alternatively, we can think of it as being how the cell manages matter and energy. And metabolism is organized into pathways and cycles. So you might have one compound which is turned into another, which is turned into another, turned into another, A, B, C, and D here all representing different compounds, and these here would be different reactions that I'm numbering. And as we'll see later, those reactions are going to be catalyzed by protein machines called enzymes. You can also have the same kind of thing organized into a cycle where the initial part of the cycle is actually regenerated as you go, and there might be things here that feed into and out of the cycle. And we'll see examples of both pathways and cycles as we progress in the course. We can also think of metabolism as being broken down into anabolism and catabolism. These are opposite processes, where anabolism is building up more complex molecules from simpler molecules. So for example, going from something like that, sticking multiple of them together to get a more complex molecule. And that requires energy. You can remember this by anabolic steroids help people build up muscle. Catabolism is the opposite. It's breaking down more complex molecules into simpler molecules, and doing that releases energy. An example of a catabolic pathway is cellular respiration. That's the process that we use to break down foods to release the energy that we use to fuel ourselves. It's breaking down more complex molecules into simpler ones, releasing energy. Whereas an example of an anabolic pathway would be photosynthesis. Photosynthesis being what plants use to harness the energy in sunlight to build up more complex molecules out of carbon dioxide and water. Let's go into this a little further. So first, a couple more terms. A polymer is a molecule that's made up of repeating subunits that are all joined together. So for example, this illustration of multiple different subunits joined together, like cars in a long train, or like beads in a necklace, or like words in a sentence, this is an example of a polymer. Each of these would be similar or identical, and the subunits that make them up are called monomers. So many anabolic reactions involve linking together monomers using covalent bonds into a polymer, and many catabolic reactions do the reverse. Let's take a look at a specific example of an anabolic reaction, condensation or dehydration. So let's imagine that we already had two formed polymers. I'll give that one 3 monomers in it, and this one will have 4. But we wanted to join these together. This one over here has a hydrogen on this end and a hydroxyl group OH on this end, and similarly over here we've got a hydroxyl group and a hydrogen. If I wanted to bring these two together and I could remove that HOH or H2O, that water, then I could put them together and get a longer, in this case, 7 monomer polymer like this. And I've still got the hydrogen on this end and the hydroxyl group on this end, but my water has been released. So this is an example of a condensation or a dehydration reaction where we are taking 2 smaller molecules, removing a small molecule, here water, and producing this longer polymer. If we were to run this in reverse and insert water in there, we'd get back to where we started from, and that would be a catabolic reaction called a hydrolysis. Lysis means split, hydro from water, so we're using water to split the molecule. So the reaction going this way is an anabolic reaction, specifically a condensation or a dehydration reaction, and that would require an energy input to make this larger, more complex molecule. Running the reaction that way, using water to split it up, would be a catabolic reaction, and that would release energy. And we'll see examples of both of these kinds of reactions as we go further. Over the next few lessons, we're going to explore the 4 classes of biological molecules, those being carbohydrates, proteins, nucleic acids, and lipids. And as we'll see, the first 3 of these, everything except for lipids, these are all polymers, where they are larger, more complex molecules made up of smaller and repeating subunits. So that's what we'll explore over the next few lessons.